Welcome, folks, to Chapter 14, where we're going to be talking about infection, infectious disease, and epidemiology. Essentially, how do those microorganisms that we know live in and on us and in our environment sometimes become pathogens within us as human hosts? So what we're going to be doing is, with the bulk of the chapter, we're actually going to be covering it in this video, in this lecture. But for the epidemiology part, I'm going to send you to the CDC's website called Solve the Outbreak, and you're going to actually interact with data related to various epidemiological cases and actually answer questions related to that. And that'll give you an idea what an epidemiologist is working with and also how different types of pathogens spread within communities. So we know that microorganisms live within and on and around us all the time. And we have different types of relationships with those microorganisms. So for example, there are at least the same number of microorganisms in and on our human body as there are human eukaryotic cells within our human body. So around 40 trillion microorganisms. And so some of those microorganisms we have a mutualism with, where we're actually benefiting one another. So for example, microorganisms that are helping with digestion or with the immune system, so on, or with our immune system, and so on and so forth. There are microorganisms that exist in and on us, but don't seem to have a clear mutualistic or symbiotic relationship. We call those commensalisms, but it is actually difficult to prove that a relationship is commensal. Oftentimes, there's some sort of benefit that we just aren't accounting for within that relationship. Now, there are also times when microorganisms, although this is less common when we compare to all the different interactions that we have with microorganisms, there are times when microorganisms become pathogenic or parasitic. So what we're going to do is start with the human microbiome, talk about how sometimes out of our microbiota there arise pathogens, and then we're going to go into the kind of outside environment and see how pathogens can enter the body that way, and we'll continue our discussion from there. So as we have interacted with the microbiome in this class as well, the human microbiome includes a number of different communities that live on the different environments that our body provides. And these include both resident microbiota that live kind of in and on us in the long term, as well as transient microbiota that might come in when we ingest food or something like that, and then move past or move on. Some of those transient microorganisms are pathogens that are actually neutralized by our immune system, and some of them could be beneficial to our microbiota. Like, for example, some of the bacteria that are in yogurt, for example, they kind of move into our digestive tract, but not necessarily all of them actually take up residence there, and kind of they can pass through. So, what are some of the kind of environments that we provide for microorganisms? We have our respiratory tract, which has different microorganisms within it, some bacteria, sometimes some fungal species as well. Really, the richest environment for microorganisms is our digestive tract because it's moist, it has a lot of metabolites. It's really a rich environment for microorganisms to live in. Has a significant diversity of microorganisms from bacteria, for example, Bacteroides, Akkermansia, Actinomyces, so many different types of microorganisms that live within the digestive tract and generally support digestion as well. There are also moist regions like the urogenital region that can have resident microorganisms, specifically bacteria and sometimes yeast as well. And then the skin and the surface of our body, including our eyes, also can have microorganisms. The skin specifically, as we have mentioned before, is more of a dry, aerobic, and saline environment. So the communities that live in and on the skin are a little bit less diverse and less kind of rich, and they tend to be more specific to those kinds of conditions. So we have these normal resident microbiota that kind of come with us at birth, either through exposure to the vaginal microbiota via a vaginal birth, or exposure to the microorganisms that are on the skin via C-section. And then as that baby grows and develops, they grows and develops with them their microbiota within the first couple of years of their lives. Kind of establishes itself in and on themselves, and that microbiome will be responsive to its environment. So different types of food, different environments, different stressors, it will change, but it tends to be pretty stable kind of after the childhood years of life, with some changes, of course. Now, speaking of stressors, environmental stressors, toxins, immune suppression, just, general overall stress within the body or stress from environmental sources can lead to changes in the microbiome. And there are organisms that live naturally within the microbiome that can also be opportunistic and be pathogens, right? And so this is very kind of, we kind of have anecdotal experience with this all the time, because in the macro world, for example, in a garden or a forest, you can have a rich, diverse community that is kind of existing cohesively together. And then you can have a disturbance, like a clear-cutting event, or, some sort of garden ill maintenance, right? And then one of those organisms can take over as a weed or an invasive species, right? And so you have organisms that are part of the normal healthy microbiota that can then be opportunistic pathogens and actually produce illness from within the microbiome, okay? So that is the human microbiome. There are also other sources of pathogens from outside of the human, okay? And that includes reservoirs. Let's go to this slide here. A reservoir is an organism or a place in which that pathogen can actually grow and reproduce. It's not a fomite or surface that the pathogen kind of just interacts with or lands on. Like if somebody sneezes onto a surface and that pathogen exists there for a short period of time, that's not a reservoir. A reservoir is actually a body in which, either a living or non-living body, in which that organism can actually exist and reproduce. So there are animal reservoirs, human reservoirs, non-living reservoirs, okay? Animal reservoirs, we know that various types of bacterial pathogens and viruses can also infect animal hosts, or they can exist within animal hosts, right? So for example, a tick that is just spreading, rickettsia, for example, between, it's a vector essentially, right? Or it can actually be a pathogen of that animal. So for example, a virus that can infect both a pig and an avian, like a chicken, and a human being, okay? And so like avian flu. And so in that way, it can exist within different species and have different reservoirs and then jump between of them, right? Humans are also animals that can be reservoirs. We call these human carriers, and these are people that are ill, either symptomatic or asymptomatic, that are able to pass that virus along to other humans. Okay. Okay. There are also non-living reservoirs, and these are soil, water, and food. And so different types of pathogens that can be spread through food. So for example, neurovirus, also soil like botulism, and then many different types like cholera, many different types of pathogens that can spread through water sources. And so, each of these kind of is unique in the way that it produces infection, and food is particularly damaging or difficult because of the ability to spread those pathogens via food, and also through a kind of industrial way of producing food, where you have movement of food from many different sources kind of mixed together. And if there's an outbreak or an exposure, it can kind of be hard to trace back. So if we have pathogens in our environment, in these reservoirs, animal reservoirs, human reservoirs, or non-living reservoirs, how do they then enter into the human host and become problematic? We have what are called portals of entry. Later, we'll talk about portals of exit when we talk about an individual who's infected and infecting other folks or other humans. And they're very similar, honestly. The ways in which we can be infected tend to be the ways in which we infect others as well. See, these portals of entry, as you might think, any mucosal membrane or moist area of the body tends to be a portal of entry or exit of a pathogen. And so the respiratory tract, and remember that the eyes also connect to that, is the most common portal of entry of a pathogen into the human body. Then also the digestive tract, and also urogenital areas as well. Broken skin or fracture, broken skin, or also punctures that actually puncture through the skin and then allow for entry into the subcutaneous layer. So, for example, thorns or needles, so on and so forth. Those are all ways in which pathogens can enter into the body. So the skin actually provides a really good kind of barrier between us and the environment. And it's very, very intricate, even though it just kind of looks like skin, but this very intricate organ that covers our body. And it has follicles and hairs and sweat glands and various layers. But if there is abrasion in the skin, that allows for entry of pathogens, potential pathogens, into the body. Now, we will talk about microorganisms later in this discussion that actually have exoenzymes that allow them to penetrate various membranes, including the skin, and actually forcibly enter the body as well. I think we mentioned this already, mucosal membranes, including the respiratory tract, eyes, mouth, nose, all of that being an entry point into the body, and the respiratory tract being the most common kind of point of entry into the body. The placenta can be an entry point, although it's less common. So we have talked previously about listeria and some other pathogens that are able to actually pass that barrier, the placental barrier, into the fetus, although that's less common. So once the pathogen is actually able to enter into the host, then what? And so when we talked about prokaryotic anatomy, we talked about multiple structures that allow for an organism to adhere to its host and also adhere to other microorganisms. So those structures then allow that microorganism to adhere to the host. And then there's other attachment factors and molecules that are even more specialized for specific pathogens, and then that can allow them to adhere to the host. And then oftentimes, as they reproduce, they then adhere to one another and create sort of like a biofilm, and that allows them, again, to increase their virulence within that host. So you can see here E. coli and its kind of attachment factors that allow it to attach to its host, and then it will actually enter the host. Chemicals and antibiotics that can inhibit entry into the host can help prevent infection, and also mutations that damage or make those kind of attachment factors or structures kind of not functional. They will actually render that pathogen non-virulent. Here you can see bacteria kind of adhering to one another and the surface of the tooth in a dental plaque. Okay, so we've talked about kind of the existence of microorganisms in the environment, and that sometimes they can use these portals of entry to then actually penetrate the host and infect the host, then what happens? Well, depending on the pathogen, you have various kind of metabolic activities that they do, the goal of which is to be able to grow and reproduce and pass itself on, the pathogen on to a new host. Okay, and so although morbidity is a potential outcome of illness, it is actually not the most beneficial outcome for the pathogen even. And so diseases that are very acute and cause sudden death are not really going to give the opportunity of the pathogen to then promote its own reproduction and kind of distribution throughout its community of hosts. So just something to keep in mind as we talk about kind of the nature of infection. So a human host will know that they're infected through both signs and symptoms. Symptoms are the signs or the impact that only the host can feel. Headache, pain, things like that. Whereas signs are the impact that others can actually measure in the host. So fever, vomiting, swelling, redness, things that are clearly identifiable and measurable. Sometimes these signs and symptoms come together, and sometimes they actually have patterns that they can then be called syndromes. But oftentimes, folks can actually be infected but be asymptomatic and still be transmitting the disease, and that can become problematic as well. And that's actually difficult from an epidemiological standpoint, to actually measure how many human hosts are infected and asymptomatic within the population. Because essentially there's no science to measure objectively. Okay, although there are tests, of course, tests for antibodies for that specific pathogen, so on and so forth. Just a note on infection and disease, this table 6 in the chapter, is really useful because it provides prefixes and suffixes that allow you to kind of understand what a disease might be related to without actually knowing what that disease is. So carcino, related to cancer. Also down here, oma is often related to cancers. Then col or colo, like colitis, is related to the colon. Derma is related to the skin. Emia is related to the blood. Endo is inside of. Gen or genesis, the beginning of, or to give rise to. Hepata is related to the liver, so on and so forth. And so you can actually look at these prefixes and suffixes and kind of know what's going on. Okay. Also, various types of causes of disease, right? You might have hereditary diseases, so that are coming via genetics, right? Congenital diseases, so some sort of difference in the way that the fetus is actually developing in the womb. Degenerative diseases, nutritional diseases based on malnutrition or different types of nutrition that lead to specific conditions, endocrine, mental, immunological, all of these that might be the cause of disease. So how do you know that it's a living microbial unit of infection? Actually, we already know the answer to this because in chapter 1, we talked about the history of microbiology and Koch's postulates, right? But we're going to review them here. So essentially, Koch's postulates allow us to go through and see, is the cause of the disease an infectious agent? And it's important to know what the cause of the disease is if you're going to be able to address that disease. And so there's 4 really kind of easily understood, but depending on the infection and the host, maybe not necessarily easily implemented postulates that Koch gives us. One is that the microorganism, the pathogenic microorganism, is present in diseased hosts, but not in healthy hosts. And of course, there's some gray area if you're talking about opportunistic pathogens that are a part of the natural microbiome, of course. The second postulate is that the microorganism that's hypothetically pathogenic can be cultured out of the diseased individual, actually isolated out of the diseased individual, and you see that isolation occurring here. And then that microorganism in the third postulate can be reintroduced into a healthy host and cause disease. And then the fourth postulate, the last one, is that you can actually then culture that same microbial agent, either bacterial or if it's a viral agent, off of that new host. Now you can see that this is a mechanism that can be potentially used, but with barriers, right? For example, it's not ethical to go through these 4 postulates with a human host experimentally. There are actually arguments that might say that even if it's a non-human host, then it's not ethical to go through these postulates, but they do allow us an understanding of how to identify if the cause of an illness is a microbial agent, prokaryotic, eukaryotic, maybe a viral agent, versus some other cause like a genetic disease or a congenital defect, or so on and so forth. Okay. So once we've identified that a microorganism or a microbial agent is actually the cause of a disease, then what? Then we want to ask a couple of questions. One, we've established that it's pathogenic, right? If it's actually causing disease. And pathogenicity just says yes or no. Does it cause disease? The second question is, how virulent is it? Virulence is a term related to the spread of disease. So if it is highly virulent, then it spreads very quickly. For example, neurovirus spreads very quickly. Okay, some types of viruses in rodents and rabbits spread very quickly, but neither pathogenicity nor virulence tell us how damaging the impacts of that pathogen are on its host. So you can have a microorganism or a pathogen that's very virulent, it spreads very quickly, but it produces a very mild illness in that population. Okay. So in terms of virulence, some of the most virulent microorganisms are the causes of rabbit fever, which is a very mild illness in rabbits and rodents. Whereas, on the other hand, Yersinia pestis is actually the cause of the bubonic plague. So it's very virulent, but it's also very damaging to its host. And then you have organisms that are not very virulent, like Candida, which is a yeast. And then, and again, that doesn't tell us about the severity of the infection or the illness that is caused. And then you have variants or variations in between. Okay. So in the next couple chapters, we're going to talk about immune defenses. So I'll leave that discussion till then. Now, let's talk about, once we actually have the pathogen has infected a host, what are some of the things that happens? What are the virulence factors, the factors that actually allow it to spread and take root within the host? And then what does illness look like over time? So pathogens will enter a host. Sometimes they'll use exoenzymes. So we've actually in lab, you have or will be looking at exoenzymes related to starch, related to collagen. And so these are some of the same enzymes that are used for pathogens to actually invade and enter the body. And essentially what they do is the release of those exoenzymes allows them to dissolve or remove the barriers. They could be physical barriers like skin and collagen between them and the body, getting deeper into the tissue so that they can actually cause infection. Okay, and so here you can see some examples of that.
One way in which pathogenic bacteria are able to create an infection and cause disease is to have certain traits called virulence factors that help them attach to or penetrate the host tissues and to escape host defenses. One group of virulence factors are enzymes secreted to help the pathogen to penetrate tissues, allowing them better access to nutrients and helping them to reproduce. Cells that make up body tissues are held together by molecules like hyaluronic acid, a polysaccharide that forms a chemical glue. Some pathogens, such as certain Streptococcus species and Clostridium perfringens, secrete an enzyme called hyaluronidase that breaks apart this glue and makes space for the bacterium to penetrate deeper within host tissues. Collagen fibers, found at the base of superficial tissues, are another barrier to pathogens. Pathogens like Streptococcus and Clostridium secrete collagenase, an enzyme that helps them to pass through the barrier by digesting collagen fibers and allowing these pathogens to move deeper into the body. Streptococcus pyogenes, which causes strep throat and necrotizing fasciitis, secretes an enzyme called streptokinase that digests blood clots by breaking down the fibrin proteins that provide the structure of the clot. For this reason, streptokinase is sometimes called fibrinolysin. Some strains of Staphylococcus aureus secrete staphylokinases that do the same thing. Both of these species of bacteria live on the skin. The ability to dissolve blood clots allows them to penetrate deep beneath injured skin and cause a more serious infection.
So you can see how those pathogens are degrading essentially the barriers between the body and the environment, and oftentimes that leads to the degradation of actual tissues as well, so that they can kind of insert themselves within the body and cause infection. Okay. Not only are enzymes released sometimes also toxins are released with the same effect, right? So you have exotoxins, toxins that are released outside of that organism, but also gram-negatives have endotoxins within their cell wall that actually kind of especially can be released when they're dying off. And that can can inhibit the immune system from successfully removing them from the body and actually kind of lower the immune defense of the host so that the infection can take on. So you can see those here.
Exotoxins are bacterial proteins that are secreted by a living bacterium into its surroundings. In a host, these exotoxins circulate and cause damage to host cells in various ways. Exotoxins can cause severe damage or death at very low concentrations. Exotoxins can be classified in different ways. One method classifies the exotoxins by the area of the host that is infected. For instance, neurotoxins are toxins that affect the function of neurons. Tetanus and botulism are caused by species of Clostridium that secrete neurotoxins. Enterotoxins, like those secreted by the cholera bacterium, affect the lining of the intestinal tract. The term cytotoxin refers to an exotoxin that kills or seriously damages host cells in general. Corynebacterium diphtheriae is a bacterium that secretes a cytotoxin that inhibits protein synthesis in host cells. Exotoxins can also be classified by their structure and function. This classification scheme has three categories: A-B toxins, toxins that disrupt membranes, and superantigens. Most exotoxins are proteins with two domains, A and B. The A domain of the exotoxin is an enzyme that has a particular function. The B domain of the exotoxin is the binding component. It binds to a specific receptor, usually a carbohydrate on the host cell. Once the B domain attaches to the host cell uses endocytosis to bring the exotoxin inside. Then, the B domain opens a pore in the endosome, allowing the A domain to escape into the cytosol, where it can inhibit protein synthesis or interfere with the host cytoskeleton. This usually results in death of the host cell. Membrane-disrupting toxins exert their effect without entering the cytosol. They can form protein pores in the host plasma membrane. Or they can disrupt the phospholipid portion of the membrane. In either case, the cell lyses. Superantigens are bacterial exotoxins that stimulate an excessive immune response. First, it causes a proliferation of T cells. Then it causes the T cells to release excessive amounts of cytokines, which can stimulate fever, inflammation, and shock. This very intense response could lead to death. The toxins secreted by Staphylococcus aureus in food poisoning and toxic shock syndrome is an example of a superantigen.
So you won't need to know all of the details of these different toxins, but you will recognize that many of them are antibiotics, right? They are antimicrobial or antibiotic factors. In this case, they're specifically related to the host, but they could also be toxins that are related to controlling microorganisms in their environment. And this is where we get antibiotics from oftentimes, right? We might modify them to make them semi-synthetic, or we might try to copy them when we make completely synthetic antibiotics. But oftentimes we're actually reaching into the toxins that are available via microorganisms and utilizing those to then respond back to those microorganisms. So you won't need to know these details yet, but when you do cover the lecture on antibiotics, then you'll start to get into these mechanisms and when we actually utilize them against microbial pathogens. Endotoxins.
Endotoxins are a part of the outer membrane of the gram-negative bacterial cell wall. The endotoxin is the lipid A portion of the lipopolysaccharide, or LPS, in the outer membrane. Endotoxins are only released from the bacterial cell when the cell dies or during cell division. When bacterial endotoxins are liberated, often during phagocytosis, they stimulate macrophages to produce cytokines called interleukin-1. Interleukin-1 is a protein messenger that travels to the hypothalamus in the brain, causing production of prostaglandins. Prostaglandins cause fever, an early sign of an infection. Endotoxins and interleukin-1 also help to stimulate phagocytosis, complement activation, and antibody production by B lymphocytes. When endotoxin is released in large amounts or within the bloodstream, it can have more serious and even deadly effects. Massive amounts of cytokines, particularly interleukin-1 and tumor necrosis factor, cause loss of fluid from capillaries and vasodilation, both of which cause a lowering of blood pressure, often to dangerous levels, leading to shock. These same cytokines also stimulate blood coagulation, which makes the shock even more severe. This deadly situation most often occurs in bacterial sepsis, caused by E. coli or meningococci in the bloodstream or spinal fluid.
Okay. So we have these virulence factors that are either enzymes or toxins, but then also those mechanisms, as well as other mechanisms, can be used to evade the immune system. Okay, either inhibit the immune system or evade the immune system. And these examples are specifically related to evading the immune system. So you could just have structural components like the bacterial capsule that allows that organism to evade the immune system, but it could also produce chemicals that actually either don't allow the immune system to engulf and actually detect and engulf it, or there are even microorganisms that will kind of live cozily within these white blood cells within the immune system, and they'll actually be engulfed, and then they'll actually inhibit that white blood cell from digesting it or lysing it. And they actually grow and reproduce within the white blood cells. And so here you can see examples of that.
The human body has many surveillance mechanisms for detecting pathogens. Phagocytes are able to detect foreign cells by looking for molecules on the surface of those invaders that are not found in the human body. Adaptive defenses, such as B cells, antibodies, and cytotoxic T cells, are looking for a specific antigen on the surface of the invaders. If a microbe can hide from these surveillance methods, then it's more likely to reproduce within the body. When immune surveillance cells, such as macrophages, are looking for invaders, they search for surface molecules that are not found on host surfaces, such as peptidoglycan. Capsules that surround some bacteria are usually composed of polysaccharides similar to molecules found on host cells. Thus, when a macrophage encounters an encapsulated bacterium, it doesn't recognize it as foreign and ignores it. This is a strategy used by Streptococcus pneumoniae, a common cause of bacterial pneumonia. Even if a bacterium is engulfed by a phagocyte, it may not necessarily be killed. The tuberculosis bacterium is able to survive within a phagocyte by preventing the fusion of lysosome and phagosome. This keeps the digestive enzymes away from the bacterium. Bacteria such as Shigella and Listeria can actually escape the phagosome and live within the cytoplasm of the phagocyte. Even if the phagosome does fuse with the lysosome, some microbes are not affected by lysosomal activity. Leishmania, a protozoan, is able to resist the lysosomal enzymes. Legionella and some staphylococci inhibit the pathway that leads to oxidative killing within a phagocyte. Some bacteria, including the tuberculosis bacterium, actually reproduce within the phagocyte. Several different types of microorganisms are capable of hiding from host defenses by changing their surface antigens frequently. These changes may occur as a result of mutations or genetic recombination. RNA viruses such as influenza virus or HIV frequently exhibit antigenic variation on their surfaces. Trypanosoma, the organism that causes sleeping sickness, has a surface glycoprotein that undergoes frequent changes. In all of these cases, it is the survival of the fittest. The microbes with the undetectable antigens are the ones that are least likely to be detected and most capable of reproduction.
Okay. And so, again, you don't need to know all of those details, but it allows you to see the diversity in the ways in which these organisms are actually kind of evading the human immune system. So once you have a pathogen, either in the environment or in a reservoir that actually enters into the body and is able to, establish itself and infect that host, then what does the illness look like? And so essentially we have these periods of that a host goes through in relation to illness. So first, when the pathogen enters the host and actually starts to take residence and generate an infection, there's an incubation period before we actually have any symptoms or sign of disease. Essentially, it's growing and reproducing and, getting to establishing itself to kind of a quantity or, the ability to actually create or to generate an illness. Then you have a series of stages between when the infection actually happens and the illness, okay? This is called the prodromal stage, and this is like when you start to feel like you might be getting sick, right? So there are some signs, but not as many, not a full-fledged illness or sickness. The illness phase is when there are actually signs and symptoms that are clear and tied to the illness. This is usually when the pathogen has established itself in number in the body and is actually producing the signs and symptoms of illness, but the immune system still hasn't built up a quantifiable or large enough response. And once that immune system actually builds up its response, then we start to see a decline in that illness, and then convalescence, and then recovery. Now, depending on the illness, these stages can be longer or shorter. So you have organisms that have very short incubation time within a matter of a day or two or a couple of days. You also have organisms that have a very long incubation period over even generations, right? You have organisms that can even live within a human host for decades. For example, Bilharzia, which is caused by a protozoa. So just depending on the illness, these phases will look different. Also, depending on the illness, decline and convalescence will look different. So oftentimes there are no signs and symptoms in this period after illness, but the person is still infectious. And we know this, again, within a pandemic, that there are times before illness and sometimes after illness that a person can still be a vector for that specific illness, but they're not showing any signs or symptoms. Okay. So speaking of being able to transmit disease, how does that happen? Again, remember we had talked about portals of exit. Portals of exit tend to be similar to portals of entry. So any moist mucosal membrane, eyes, ears, mouth, nose, urogenital areas, abrasions of the skin, digestive system, these can be ways that pathogens enter the body. And oftentimes the way they enter tends to be the way that they're also reciprocated or infect others, although there can be differences. Okay, and so transmission can occur through contact with the individual that's sick, either symptomatic or asymptomatic, or between contact with the individual that's sick in a fomite or an inanimate object that they actually interacted with, right? So when a person sneezes or coughs onto an inanimate surface, and then some pathogens can actually exist on that surface for a period of time, usually not very long, but a period of time, and then another person can actually pick up that pathogen from that surface. You could also have a vector involved, which is another living organism that actually moves the pathogen between different hosts. And so this kind of gives us an idea of all of those different types of transmission.
Infectious diseases are transmitted from either a host's portal of exit or a reservoir to a new host. There are three major categories of transmission: contact transmission, vehicle transmission, and vector transmission. Contact transmission is the spread of pathogens by direct contact, indirect contact, or droplets. Direct contact can include touching, kissing, or sexual intercourse. It can also include transfer from mother to fetus, and contact within a single individual, such as between the nose and the eye. Droplet transmission occurs via mucus droplets within a distance of less than 1 meter. This can occur while exhaling, speaking, coughing, or sneezing. Indirect contact involves spreading of pathogens by inanimate objects called fomites. Fomites can include needles, eating utensils, money, tissues, toys, and medical equipment. The second major category of transmission is by vehicle. Vehicles can be air, water, and food, as well as bodily fluids such as blood that are handled outside of the body. Airborne transmission involves the spread of pathogens via an aerosol. This aerosol can be either dust or droplets traveling farther than 1 meter. Waterborne transmission is a major mode of transmission for many gastrointestinal diseases such as cholera. This usually occurs when fecal material enters the water supply. Foodborne transmission is also typical of many gastrointestinal diseases. The pathogens may enter the body in food that is improperly prepared or contaminated with fecal material. Vectors are arthropods that transmit diseases from one host to another. The term vector is most often used to describe insects or arachnids that are part of the pathogen's life cycle. For instance, the mosquito in malaria or the flea in bubonic plague. However, some insects can also passively carry pathogens with their bodies. For instance, a fly may carry typhoid or dysentery from a host species to the food of another person.
So here you can see those different modes of transmission in action. Okay. So the book will continue through, well, actually, we could just go back to that section there. The book will give you more details on each of those modes of transition, but that gives you an overview of all of them as well. So what I would like you to do is after you close this video, go ahead and review your notes, and then go into what we're going to be doing is the epidemiology section. We'll be doing it in an active way rather than just through the lecture. And so you're going to go to the website that's below, but I'm also going to show it to you here. And it's the CDC website, and it's called Solve the Outbreak. And it's an activity that allows for you to really kind of look at the transfer of pathogens in action. Okay, I had to mute myself for a minute because the significant amount of a sound that was involved in that. And let me just redirect. Okay. Well, hopefully you can still hear me, but for some reason sharing is not working. I'm not sure if maybe the website or the different graphics are just too much for the sharing mechanism in Zoom. So what you're going to see is that you're going to be asked to select from two different levels, level 1 and level 2. Level 1 is the only one that's unlocked. So go ahead and go into level 1 and you'll see a number of different options. And you'll go into one of those options and you'll go through the different graphs and the different questions that are related to those graphs. And what I want you to do is to go through 2 of the outbreaks and go through, complete the outbreaks, and then move into the Jamboard that's linked below and let us know which outbreak you did and kind of a summary of what happened within the outbreak and how you followed through and figured it out. Okay, I'm actually going to close this website so it stops interfering with my Zoom. So thank you for listening. Enjoy the CDC Solve the Outbreak activity. Make sure to report back to that Jamboard for points, and I'll see you in the next lecture.